Hi guys, Dr. Orlando with a video on cell cycle. We're going to go through the phases. We'll go through the phases of interphase in detail, and then mitosis I'm just going to name them because I'm going to do a separate video just for mitosis. Alright, so cell cycle are all the events that happen to a cell during its life. And there are two phases to the cell cycle. Shown in blue here is mitosis. Okay. And shown in orange is interphase. So this whole area here is interphase. A lot of things happen during interphase, and so we're going to spend some time there. Okay. Mitosis is nuclear replication or nuclear division and cell division in the end. Okay. And then interphase is everything that happens between the mitosis. And most of the time when you look at a cell, it will be in interphase, doing its job or preparing for mitosis. So let's look at that. So we'll start with a cell that has just gone through mitosis. So we're going to start actually right here on the circle. Okay, this cell has just been made by mitosis. So the first thing it's going to go into is G1 or gap 1. So during gap 1, the cell is going to grow because it just went through cell division, and it's smaller than it will be when it's grown up, okay, when it's mature. So it's going to grow, gain cytoplasm, make more organelles as needed. Okay. It's also going to specialize. And it may differentiate. That means it's going to become more specific. It's going to get its job, as I said in class. So a cell that is more generic might be put down a path to become a red blood cell or put down a path to become a muscle cell. Okay, so it gets its orders. And the way it gets its orders is that there are signals that come to the cell and tell it to start using specific parts of its DNA to grow specific parts that make that cell unique. So it's getting its job. Okay. It also can become sensitized to external signals, so it's going to grow receptors. It's going to, let's not say grow, let's say make receptors. It's going to make receptors to receive those signals to tell it what to grow. Okay. And it's going to, at this point, have an option. If this cell in the future will go through mitosis, then it may continue on in interphase. If it's done, it has been made, it got its orders, it's going to go do its job now, we say that that cell is then committed and it's ready to go do his final job. He's mature. He's not going to go back through mitosis. So those cells would go into G0. In G0, or really G0, in this phase, the cell is now amitotic, which means it's not going to divide anymore. No more division. It's just going to do its job. Okay, it's a mature cell. It's going to go do its job. Now you see this arrow going back in. So occasionally cells can come out of G0 and go back into G1 if they get the proper signaling. But most of the time, as we understand, if the cell's over there in G0, then he's doing his job. Okay, and he's do his job until he dies. Okay, so that's G G1 and G0. If a cell in G1 gets a signal to divide, like for example, if a growth factor or a hormone is sent to that cell that tells it it needs to grow, it needs to divide, then it needs to prepare for cell division. So it'll move on through interphase, and it will go to the S phase of interphase. In the S phase of interphase, there is, S stands for synthesis, and this is where DNA is replicated. Just like we talked about in class, where one molecule of DNA is copied and we end up with two identical strands of or molecules of DNA that are then organized and paired into sister chromatids. Okay, so the DNA is replicated. We're preparing to divide the cell. We need to make a copy of our library. All right. From there, the cell has copied its DNA and needs to move on. It's going to go to G2. Okay. And in G2, this is the second gap, or gap 2, okay, second growth phase, the cell is going to grow some because it knows that it's going to divide. And if it's going to divide, it needs more cytoplasm, more room to put all of these new chromosomes in when it makes a new nucleus. Okay, it's also going to make stuff that it needs for cell division. So it's going to make spindle proteins. These help move the chromosomes around during mitosis. It's going to make its second pair. Zoom in a little bit so I can write neater. Second pair of centrioles. Centrioles will be like the base or the motor for moving the chromosomes around. Okay, so it's growing and it's making the stuff that it needs, the spindle and the centrioles, that it needs to go through mitosis. Okay? All right, so then, go over here. So then, once everything is made, we've copied our DNA, we've grown and made our spindle, now it's time to go through mitosis. Let's zoom in a little bit here. Now mitosis is made up of multiple phases. Okay, so the first phase, and again I'm not going to go through the details of these because we'll have a separate video on it. Okay, the first one is prophase. Metaphase is the second. These are the phases of mitosis. Prophase, metaphase, anaphase, telophase, and then after that, nuclear division is actually done. Two nucleuses will be in one cell, but our goal is to divide up the cell. So the last thing that has to happen is not really a phase of mitosis, but it's required for cell division, and that is cytokinesis. And that is cell splitting, where the cytoplasm actually is split so that we have two separate cells. From there, once that cell goes through mitosis, well then it's going to continue in the cell cycle. So this would be the two cells that were made. Give them each a nucleus. Okay, it can continue on through the cell cycle, and then this guy's going to follow his own cell cycle and continue on, either going into growth phase and copying himself again, or going off and doing his job. All right, so that's the basic idea behind the eukaryotic cell cycle. Should mention that. I don't think I said that. This is eukaryotic. Let me spell that right. That's better. Eukaryotic cell cycle. We know it has to be eukaryotic because eukaryotes have nucleus, and the nucleus is what's being divided in mitosis. Okay, if you have any questions, please feel free to contact me. Have a great day.